Real engineers. Let's get a couple. Cognitive neuroscience. Medicine. Neurology. Okay, good number. I'm trying to get an idea of sort of the general background for people in the audience so I know sort of how to pitch the talk. Because the idea is to give, my job is to give you, enough background so that as you go through the rest of the day, you have sort of a tethering point so that you know when things are talked about what we're talking about. Now, since this is a workshop, it's quite important that you feel empowered to ask questions frequently. So don't be shy. If there's something I say that makes absolutely no sense, to ask me a question. And I'm happy to answer that. And if I can't answer that, I'll pass it off to one of the other people in the room. But before I do that, wave. See, hi, Mom. Hi, Mom. Hi, Mom. Excellent. I'll send this to your moms. That way she'll have copies. So what I want to try and do is, Petra gave a very good motivation for the virtual brain in terms of the general framework. I want to give a bit more in terms of the theory around why you want to do modeling at this particular level. And then I'll blast through the platform itself, talking about the architecture, how we incorporate dynamics, how we incorporate the spatial temporal structure of the brain, and then a bit about the simulation platform as well. There'll be more. This will be repeated again and again throughout the day. So by the time you leave here, you will hate the word virtual brain, but you'll know how to use it. So the notion is that the brain works through the interaction amongst elements. The brain works through network operations. And there's a number of ways that you can interrogate brain networks. You can collect data yourself. You can grab data from different platforms. There's tons of data out there now. Human Connectome Project, for example, has thousands of data sets. UK Biobank has thousands of data sets. There are a number of data sets out there that provide you a fairly comprehensive picture of a person. So you can get databases that have genetic information, they have structural information from the brain, functional information from the brain, phenotypical information from the brain, etc. You can pull all this stuff together. The challenge is really how to understand that. Now, you can look at the individual elements. You can look at the genes by themselves. You can look at the structure by themselves, function by themselves. People do that, right? You see papers like that all over the place. They analyze functional connectivity, for example, in the brain and look at that only. And that's looking at a piece of a puzzle. You look at the needles, for example, on a leaf, or you look at the branches on a leaf. But you miss the fact that for that organism to operate, it really requires the conjunction of all those pieces. To understand its interaction, to understand how it manifests in the real world, you really want to try and put it together. And that's the notion behind computational modeling, is to give you a way to pull together disparate data sets to try and understand how their combined operations work. This is a schematic for the virtual brain platform. The notion here, again, is to try and capture structure and function and how the unification of those two elements produces brain operations. I'll go through this slide again a bit later on. You'll see it again at least one more time during this talk. So the idea is to not look at individual elements, but actually look at networks. So you can have cell populations, which are the neural masses that we're going to be modeling today, their interactions. You can also have the distal connections between areas of the brain, so long-distance connections. Those long-distance connections are estimated using diffusion tensor imaging, for instance, and you'll see that a bit later on. It's too bad it's so dark up there, isn't it? Anyway, there are examples of large-scale brain network modeling in the literature. It goes quite far back, actually. There's examples from Paul Nunez a long time ago in the 70s, looking at homogeneous connections between different elements, and then Victor Yersa, Peter Robinson, and Combs looked at introducing some inhomogeneities in the platforms. And then our group started looking at virtual brain in terms of heterogeneous connections, so you have time delays now, basically, between elements. And those time delays introduce heterogeneities in the model, and that produces then interesting dynamics in terms of how the systems evolve. And then a bunch more papers that kicked off the virtual brain in its current manifestation. I'll say it again. A lot of these papers you'll be able to access through the Virtual Brain website. So if you go there, there's a list of papers that goes back to about 2004, I think, actually. Some of them, at least. So you can grab those papers from that website, or at least the references to them. Where's my wine? Right there. Okay. Cheers. It's often the case that when people use the virtual brain, you will get asked what the relationship is between that platform and other modeling platforms that are out there. The one that's probably the closest is the modeling platform that's done in the Human Brain Project, and that's called Blue Brain historically. But there are other modeling platforms that approach modeling in a slightly different perspective. In some ways, that's captured in artificial intelligence, machine learning, computer science, and sort of the traditional framework, where you build algorithms that are meant to capture aspects of brain function as far as you can conceive them. And the intention there is to put together these functions under the hopes that that's how the brain does it, or at least captures an aspect of how the brain does it. And that's things like, there's a platform called Nengo by a colleague of mine in Waterloo, Ontario, Chris Elias-Smith. And his manifestation is called SPAWN, which basically links cognitive modules, if you will. So working memory, motor function, language modules, so on. Put some modules together with the hopes that that reflects some aspect of how the brain does it. Doesn't necessarily incorporate biophysics, doesn't necessarily incorporate anatomy, but it incorporates function. So it's almost like a top-down approach. You build a function and hope that's how the brain does it. Not a bad thing, but that just tells you exactly what the level of inferences are that you can derive from those models. Our approach is more of a bottom-up approach. Now, this is an example of a really, really bottom-up approach, Blue Brain Project, where you go and try and model individual synapses, and then synapses between dendritic axons, dendrite interactions, whole cell ensembles, and then put the ensembles together, et cetera, et cetera. So you're trying to build everything from the bottom as far as you can go up. They don't do molecules, but they do synapses at least. And the notion is you build sort of critical building blocks, you put them all together, and then at some point you'll have a brain emerge from this whole thing. There are problems with that. We can talk about that later, but it is a difficult model to realize because of the fact you're modeling a nonlinear dynamical system. So the nonlinearities cause a problem for how to actually put things together, because the combination of elements is not necessarily a linear function or additive function. It could be quite multiplicative and so on, leading to very weird sort of operations that happen as you put these modules together. Virtual brain is sort of in between there, and the notion there is that we are interested in network operations. We are interested in large-scale network operations. So to model that, we want to focus on that scale, on the scale of cell populations, on the scale of connections we can measure with DTI. That's the focus of the virtual brain. Let me just go through the software platform. As you already know, most of you probably have, or hopefully have, downloaded the software and are playing with it to your heart's content. The idea, again, is to try and use information from empirical data. So you've got up here structural information that tells you the distance of connections and some estimation of the strength of a connection derived from diffusion tensor imaging. There is a default connectivity matrix we'll talk about in a minute that provides you with sort of a canonical connectivity platform where you can supplement that with diffusion tensor imaging as well. You locate regions of interest. That can be done in terms of very sort of coarse regions of interest, for example, 72 ROIs that are sort of placed across the brain. You merge those two together and then generate dynamics. And you can generate time series from local field potentials all the way up to EEG, to fMRI bold, and look at higher order measures of those data. For example, functional connectivity here. This is the correlation of time series that are simulated in the platform. When you go to the website, you'll see that's the first page that comes up. We've done really well with the downloads recently. We've up to, this is a couple days ago, over 10,000, 700 in the last 30 days. So it's not just relative, that's actually people, unique people downloading the Virtual Brain platform. In here, there's publications listings here that you can grab, and it'll give you the list of publications. If they're open access, you can grab them directly, which is nice. The download window's there. There is a lot of help online, which is nice. There is this documents, docs.thevirtualbrain.org, that has some tutorials. You probably have grabbed those already, hopefully, or at least seen them. And as we develop our workshop, we will take feedback that we get from you all and incorporate that and improve the documentation. So part of your job here today is not just to learn Virtual Brain, but tell us how we can do this better for you next time we do it. It's an evolving process. This is the docs platform there. You can also obviously grab it through GitHub, which is a more direct, and also allows you to access the command line version, which is, for those of you who are facile with Python, is a nice way to work with Virtual Brain. That's just meant to confuse you, so look at that, get confused, and then we'll move on to the next slide. Good, okay. This is a less confusing slide that, again, gives you the quick overview of the workflow. So the notion is to get information from individuals or groups and put them together. The spectrum of information can be quite variable across studies, of course, because you may only have one particular data set from an individual, but the idea is to try and get as much as you possibly can. So in this example, we've collected data from a newspaper reporter and measured her functional MRI data, her structural data to get her geometry, as well as using diffusion tensor imaging to do tractography on the person as well. Fed that through the simulation platform, and then generated simulated bold data. It's on the top, and this is the real bold data on the bottom. The patterns are similar, though there's a time lag in terms of, if there was a movie, you'd see that there's actually a time lag between the two. But that's done on one individual. And that's really where I think where Virtual Brain is unique and is quite powerful in the sense that you can generate models for individuals, which you'll see later on when we start talking about clinical applications. The utility of that, of course, is to be able to use that potentially as a way to help diagnose individuals, potentially also use that to guide interventions, whether epilepsy, stroke, or dementia, for instance. When you open up the GUI version of Virtual Brain, there's a number of tabs that guide you through the different functions. The first one that comes out will be a user one. Projects will give you sort of the broad outline of what's in your database. And then the simulator is the main thing you're going to be using to generate the simulations, oddly enough. So there you've got the selections for the different kinds of models, the features of the model, and then the output that you're going to be generating once you run the simulations. One of the there. Is it moving? Anyway. So this is the connectivity matrix. This is one of the features of Virtual Brain. What you see here is an adjacency matrix. Adjacency matrix is a matrix of connections between different elements. You've seen this a lot if you look at fMRI or structural MRI, it's DTI, I should say. So this connectivity module allows you to look at the connections that you're going to be using as the foundation for your model. Regions of interest, the connection, this is the connection, I think this is the weight, yeah. You can also change to the lengths, the distance estimates as well. Get some graph metrics, that's what the size of the balls has to do with. You can use this to introduce lesions as well. So you can go in there and edit out, for example, connections, edit out regions, and introduce sort of virtual lesions, which I think we're going to be using for the stroke example later on. Once you've done that, you can generate the simulations. This is a bold simulation, fairly coarse regions of interest. Navigations, you'll see that the colors are changing on the brain. You can change the granularity, how it's shown. A lot of this can be saved as well for use later on. ROI placements, etc. That's bold fMRI. The next one is EEG. What you see here is the dural surface with the locations of channels and the sort of field of view of each of the channels in the forward solution for the model. These are the different channel time series. And then in a second, you'll see the placement of the electrodes on the head. This, again, is a default configuration. You can add in your own as long as you have the locations of the channels. There's also one for MEG in here. Well, I can't remember how many channels is in MEG, though. It's like 200, I think, something like that. 256. Anyway, those are all configurable as well, but there's a default in the platform. And there also is a series of analysis tools. Some of these are MATLAB plugins. Some of them are directly written in Python. We don't have a lot of analytic capacity in the platform, and that was done on purpose because the notion is that you'll generate your simulated data, and then you'll analyze it however you want. And so we provide some basic functions that can be used, for example, for optimization and quick checks, but we don't have a lot of embedded functions. You can generate those yourself as well. Since it's all written in Python, you can add in as you wish going forward. Let's talk about some math stuff, shall we? Since it's early in the morning, it's a perfect time to talk about math. Okay. The approximations, the dynamics in virtual brain are generated through what are called mean field approximations. So instead of modeling individual cells, which you might find in a Hodgkin-Huxley Morris-LeClaire model, we're actually doing mean field approximations. So basically think about it as generating the activity of a population of neurons. It could be several thousands, could be several million neurons. There are various flavors of that. I think there's 14 different versions now of mean field approximations in the current version of Virtual Brain. I'm not going to go through them today. Thank me for that. I'm thanking myself for that as well. But there are different kinds. You've got, for example, local field potentials, which is the Stefanescu-Yersa flavors. There's a Wong-Wang model. There's also a reduced version of that, which you might hear about later, that Gustavo Deco has been using. Firing rate models, all the way back to Wilson-Cowan, Brunel, Wang, Janssen, Ritt. And then what are called phenomenological models. They basically generate time series that represent certain phenomena without necessarily focusing too much on the biophysical elements. It's just capturing a behavior that's important to generate network dynamics. Simple 2D oscillators, a Kerr-Moto oscillator, and then the epileptor, which is being used for epilepsy research, oddly enough. So as I said, all of these are meant to capture the operations of a chunk of tissue. The size, of course, depends on the granularity of the model, but they're all can be described as nonlinear dynamical systems via sets of differential equations. This is a great diagram that kind of captures the broad perspective of what happens in virtual brain in terms of the model. There's a generic equation that captures most of the approximations that you can do in the virtual brain. And you can show this graphically as well as looking at some of the equations. So on the top of that, You've got the change in the cell population activity with the population activity itself at the time, its interactions with adjacent cell populations, and then the interactions from cell populations further away. So long-range input, short-range input. This is an input term if you want to do stimulation, for example. You would use this term here, and then you've got the local noise term. Could be Gaussian, could be nonlinear, multiplicative noise, I should say, as well. Most of the models will have compartments for excitatory and inhibitory pools of neurons, with the exception of the phenomenological ones, which tend to have one or two compartments, and maybe not necessarily cell types, but just sort of phenomena. And they will have interactions between them. This is meant to represent the Stefanescu-Ursa 3D model. There are other variations, for example, having an additional inhibitory modulation. So you've got pools of excitatory inhibitory neurons within a cell population, the adjacent interactions here, captured in a chunk of tissue, and then you've got then the long-range connections that you capture from diffusion tensor imaging. All the parameters in these models are modifiable, and the modification will change quite dramatically the behavior of the systems. And this is a plot of using Stefanescu-Ursa 3D model and changing the variance of the parameters. And by changing the variance of the parameters, you get quite different behavior. If you have tight variance, you've got a very uninteresting model that just kind of sits there at a fixed point and doesn't do much. If you increase the variance, you've actually got two attractors forming. See if I can play that again. So low variance, the cell collapses into a single fixed point. Higher variance, you've got the activity moving around a much broader phase. The important thing here is the fixed point's the same. What's different between these two models is the variance of the parameters. And that provides you with a much more elaborate way of capturing the dynamics. You can graph this. You'll see in a minute how to do that, and it's actually quite helpful to be able to characterize when you change parameters, what's the model going to look like. So it's a way of sort of testing what you're going to see going forward.
Sorry, that red dot is the mean field, is the center of.
It's the mean, yeah. And the dispersion, of course, is different. That's the point. Oops. Okay. So this is a criticism that comes up a lot, that if we're doing this approximation, are we actually losing information about how the brain works? I'm saying how the brain works in quotes because we actually don't know what the answer to that question is. But the idea is that if we lose some details, if we're not going to model individual synapses, do we lose information? Yeah, of course we do. But the question is that critical to understand some of the phenomena that we want to try and model? Do we capture the qualitative features that we want to actually model by using these approximations? Can we capture important features? And the answer is yes, we can. Here, what we're showing is a very simple model that Victor and Roxana Stefanescu put together to demonstrate the feasibility of using these mean field approximations. With a very coarse model here, which would be sort of the mean field approximation we're using in virtual brain, and the more detailed model with individual neurons here, local connections as well as long-range connections here. Up here on the top is a phase-based plot for dispersion and one other parameter. Where's my pointer? Dispersion and time delay, I think it is here. And the variance of the system across this parameter sweep. For the full model, like this, and then for the reduced model where you're doing the mean field approximations. The important point here is that while the quantitative features will be different, the qualitative features are quite similar. So it does capture the qualitative behavior that we want to try and understand by using these models. If you, for example, in this case, if you introduce a Alzheimer's disease by eliminating units, by degenerating neurons, this is what happens in the full model, And this is what happens in the reduced model. So again, the details, the quantitative features are different, but the qualitative features are quite similar. So the idea is that we're going to miss some details, no question, but we want to make sure that we understand what the qualitative nature is we're trying to capture in the model and make sure that that in fact is captured, and that's really the target. What are you trying to understand in terms of large-scale networks?
What do you mean by quantitative?
Quantitative, the actual numbers themselves, like where do these bifurcations occur? What specific value?
But in the reduced model, you don't see these bifurcations.
No, the quantitative features are different. The qualitative features are very similar.
What do you mean by the qualitative?
The patterns you're seeing across here. So it's the fact that there are these different segments in there. So we're going to miss some of the quantitative details, but the qualitative features are still going to be captured. Okay, I'm going to go through some just high-level examples of model instantiations and then using the phase plane plots within the virtual brain platform. Are you going to talk about this at all, Misha, in these things? Okay. When you build a model in virtual brain, you do have access to these phase space plots that allow you to look at the variables in the equations. In this case, is the inhibitory and excitatory state variables for the Wilson-Cowan approximation, and their values across a particular set of parameters. If you click in points here, you'll generate trajectories that will say, if I start here, for example, in the phase space, where do I go? Go to that attractor right away. If I start over here, I'll do a different kind of course to get to the attractor, which is where these points intersect on those lines. This is really, really helpful to understand the behavior of your system. You can modify these parameters, you can modify the scales, and that will then allow you to change the trajectories as you go forward. So with a slight shift to the parameters, for example, I go from a very uninteresting fixed point to now more of an oscillatory mode here. And this is by just changing the state variables, excitatory inhibitory balance, changing the state variable relationships, and then I get a nice oscillation. So that tool is quite useful in the virtual brain. You can do that when you, in the simulator platform, you click on regions and it'll bring up that particular tool. It's also shown here in a bit more detail. It was shown there. Well, that's interesting. So I guess there's some ghost in the machine that deleted the slide in real time, which is kind of interesting. Anyways, had you seen, you would have seen on, let me just go back to this other one. Apologies for that. This is the problem with having a 10-year-old Mac Air that gets getting grumpy as it gets older and deletes things. As it has dementia, I guess, it kind of eliminating slides. What you would see is on the sides of these windows, there would be sliders that allow you to change the parameters in those models in real time, and then reconstruct the phase space for the for that particular modification. The Wong-Wang model, it's firing rate. There's no oscillations necessarily in there, so you'll see that it's generating initial transients. This is firing rate in hertz. This is firing rate for different channels and local field potentials, and these are the phase space for threshold functions and time variables. And again, if you change that to, damn, another slide missing. My computer's dying. This is a generic 2D oscillator. This is quite a useful model for generating large-scale dynamics very simply. You can use this model to generate functional connectivity until the cows come home. Because all it does is oscillates, or it doesn't oscillate. And depending on how you change the parameters, so for example, here are the two parameters for V and W, state variables, and then the trajectories. In this space, you've got not much happening here. You have an initial transient, and then it converges to the fixed point. Whereas here, you've got a nice oscillation in the blue trajectory. So if you click here, you'll get nice oscillations in the time series. V and W are state variables again on the top there. And you can change the noise to give it a more interesting trajectory, but this is how you generate very simple oscillations. You can do more complicated ones as well, but this is a nice simple model to give you a generic platform, that's why it's called generic 2D, to generate large-scale dynamics. For example, here, we'll come back to this one later. This is actually a stimulation parameter. We're using 2D oscillators to look at the propagation of stimulation at different points in time. So the stimulus is here, stimulation, and you've got, not much happens when there's no noise, but when there is noise, you see some propagation of the stimulation. We'll come back to this example a bit later on. And the important thing, these are the sliders, for example, I was mentioning earlier, to be able to change the different parameters and look at the change in phase space. The important thing is that these mean field approximations are not by themselves. They're embedded into a network, and the embeddedness of these mean fields is what produces the interesting models that we're going to be looking at later on. And so this is an example of a couple hundred oscillators that are uncoupled, have weak coupling and strong coupling. And you'll see that the trajectories start to dephase as you add coupling because that introduces time delays. And you get much more interesting dynamics than in the large-scale model when you have both dynamics locally, but also time delays in how they're interacting. We'll get to the time delay point in a moment.
Sorry to ask.
Yeah.
Some of those models you explained, I remember formulating the noises editing.
Yes, in this one.
Is there implementation for other type of noise, like state dependent type of noise?
Not in the oscillator model. I think are there implementations for additive or multiplicative noise? I don't think so right now. There is. Which implementation?
You can choose for multiplicative noise, and then you set two coefficients. It's like A times the variable plus B.
Okay.
So there's multiplicative and additive component, and you can decide for the value of the multiplicative component and also for a time correlation.
Ah.
So I actually think you can define whatever equation. You can add an equation of yours.
Sure. Yeah, you can add it in, of course. It's just a question of whether it exists, and it does exist for some of the implementations, but not all of them, I think. Is that right?
Yes. There is an easy way to define this equation, or it's by default.
Okay.
This simple equation is very easy to set.
Yeah. This is why it's a good collaboration, because when I don't know, I can ask someone else, and they say, "You're wrong, Randy." Okay, good. I'm fine with that. Quick thing, time check. So what's my... I've been talking for 32 minutes and 30 seconds so far. Okay. Very good. Okay. Geometry is important because we want to make sure we capture the spatial distribution of nodes. So that can be captured through regions of interest templates. There is a default one in Virtual Brain that's in the Montreal Neurological or MNI space. So that's the ROIs in the cortical surface, and then the locations of the centroids in the boxes there. So region-based modeling, we've done as few as 30, and have gone up as much as 200, maybe even more now. I can't remember the last count we had for those. The main thing for region-based modeling, of course, is you're not looking at the spread from a region. So regions tried to capture all of the dynamics that are happening in that local tissue are sort of lumped into a single region of interest. You can do surface-based modeling. It's much more computationally intense, but it can be done, and it is worthwhile doing if you want to really capture the balance of local and global dynamics. There, we've done up to actually more than 140,000 nodes. I think the last one was 200 and some odd, I think it was, nodes that were used. More than that?
I did one test with
1
1 million.
million. Do I have 1.1 million?
But it was 2,000 Teslas running in parallel. It's quite intense.
Dude, Okay. So yeah, you can do surface-based modeling. It's computationally intense, but it does give you some additional information about, again, the balance of local and global interactions in the brain. The connectivity, again, is the important aspect of what happens in the virtual brain. And in the platform, you have the capacity to specify the nature of the local connections between the adjacent cell populations, as well as the nature of how the time delays manifest in the model as well. So again, the time delays are imposed by actually using the geometry and the distance the connection must traverse to go between two areas of interest. You can set the nature of the coupling convolution for the local parameters, local nodes, Gaussian, Mexican hat, and various versions of that as well. So what happens to the connectivity as it drops off from a given node. And that's just showing the same thing graphically as you have the focus here, and this is just a Gaussian drop-off in terms of the connection interaction between that node and its adjacent partners. This configuration introduces what's a really important feature of virtual brain called the space-time structure of the connectivity matrix. Now, it's different for different models, and it's worthwhile looking into that in terms of how the time delays actually manifest. So when you look at the particular instantiation, it'll have the capacity to include time delays of different magnitudes. So in the time delays, we calculate that based on the track length or the Euclidean distance, depending on the implementation as well, between adjacent points. But the time delays provide that very interesting richness of architecture on the system. This is captured quite nicely in this diagram. You can generate these in the connectivity module in TVB. So as you implement an adjacency matrix, you can look at a space-time structure and capture that directly. What this represents, and I quite like this because it does convey a very important feature of the brain that we're actually modeling. And that's the fact that even though you have this connection, the distance the connections have to traverse are not the same. And that, by design or by necessity, sets up a differential delay between the information transmitted between two nodes. So here we have a 38-node adjacency matrix. White squares indicate the presence of a connection, black indicates none. So you typically will see this kind of adjacency matrix in publications. If, however, we look at the distance the connection has to traverse to make that synapse, if you will, and introduce that as a delay, you can extrude this matrix or sprint it out into 3 dimensions. So this tells you that at 2.7 milliseconds, these are the connections that can be engaged, given the adjacency matrix I've used. At 10.7, these are the connections. At 18.7, these are connections. And at 26.7, those are the connections. So that gives you information about the potential for which areas can be interacting. It doesn't tell you that they are interacting. It just tells you how long it would take for an impulse to travel between these two areas. So it gives you information about the space-time structure of the connectivity, which is critical in how the patterns at the network level manifest, both in terms of activity, but also more important in terms of interactivity. You can stimulate individual nodes in the virtual brain. Andreas has some great experience in that. He's got some very nice work in there. This gets back to some of our earlier examples. This is killing me, man. Oh. Give me a pen. I'll just draw on the wall here. So here's a time series with a spike here. That's the stimulation parameter. It's a square pulse. Can you see that? Square pulse. At the square pulse, this area gets stimulated, and then it propagates. And if you add noise, the propagation actually is improved because the noise helps to carry the signal further, which you see here. From here, stimulation here, and then it traverses quite nicely. Cool, isn't it? Yeah. And this is, I think this is actually from you, Petra, right? This particular slide. Okay, just say yes. This is representation of stimulating different nodes in the visual cortex and looking at the spread of the stimulation. So it tells you where the stimulation happens and then the propagation of the stimulation based on how you've modeled the spread of the stimulation in the surface-based models. The algorithms for integration, there's both deterministic and stochastic. Deterministic is quite helpful for initial simulations just to get an idea how your model's working. But if you want to get reasonable data, reasonable in the sense of mapping it back to empirical data, you want some sort of stochastic algorithms. I think there's another one on here that's not here right now. The Hoyne stochastic one seems to be the one that's most well behaved right now. But if you're facile in modeling, you can use the other ones at your own risk, of course. Monitors, those are meant to convey what sort of data can you generate using the virtual brain. So since we're modeling cell populations, whether it be spiking neurons, whether it be phenomenological activity, what you're actually generating is essentially local field potentials, which is nice because that means you can generate any kind of data you want if you know what the forward solution is, going from the local field potentials to X, X being your method of choice for the simulation. So we have the raw signals from the neural population models. You can actually also capture the state variables themselves. And right now, we have forward solutions for EEG and MEG, for bold fMRI, and for stereotaxic EEG. It's quite helpful in epilepsy modeling. So as I said, the raw signals are essentially local field potentials. You can read that. It's basically just saying that what you save depends on the model. Not surprising. EEG and MEG, forward solutions, those are locations of dipoles in their various orientations along the cortical surface. EEG forward solution, MEG forward solution, they're at tangents because of the different biophysical properties, but again, you can use those forward model you can use your own forward model as well. There's one in there that has I think a 4-compartment forward solution. For the bold monitor, we're using the reduced balloon Windkessel model, which is basically looking at the conversion of the neural response to a hemodynamic response, and then generating the sluggish bold response that's now ubiquitous across the fMRI literature. And that's what generates the functional connectivity matrices that everybody's using right now to analyze large-scale networks in various species. And finally, we have the stereotaxic EEG, and this is meant to indicate the location of electrode points by implanting needle electrodes that are used to measure the activity around those needles. So you'll see examples of that later on, I think. Okay, last slide. Like most software packages, you can generate garbage in terms of the simulation outputs. And you may not always know it. And that's true for any data analysis you do. So it's quite important to really do reality checks as much as possible when you see, when you have a model that works, check and make sure it's actually looking reasonable in the sense of the time series itself it generates, but also making sure that things converge. So there are a number of things that are common, but it's always sort of going back and saying, does it look reasonable in the sense of what I see in terms of time series, for instance? Is it showing oscillations and then quiet, which doesn't really happen in the brain unless you kill somebody? So it's generating oscillations across the entire time series, that's good. But if you have an initial oscillation and it stops, that means you've missed something in the model. So coupling functions are part of the way that can happen. Integration, we mentioned earlier about the difference between the different stochastic versus deterministic integration. There's possibilities that it won't convert, and you will get error messages on that. And it's just kind of going back and making sure that the parameters you specified are within a reasonable range for that. So for example, if you've got too tight a coupling, that can actually mess up the dynamics to the point where it won't converge because it can't get a different, unique solution in the integration term. But again, as you do this, you'll get a feel for where things will go right and where things can go wrong. The tutorials that are online that you can download, it's worthwhile working through those in some detail just to make sure you can reproduce what you see there. And that helps you at least get, again, a flavor for how the program works so that when you have your own data, it's much easier to do that translation to make sure that you can model what you're interested in modeling. And again, as we go through the day, you'll have more examples of how that can be instantiated both for generic applications to looking at young subjects, old subjects, stroke, epilepsy, stimulation, and so on. So with that, I will stop now, take any questions, and then we get a break. No? I get a break. I get a break. Okay, any questions before I pass over to Misha? I know it's a lot, and again, I wasn't meant to give you all the gory details, but at least to give you an idea of how this platform works so that as we go through the day, you'll have heard this at least once, and maybe heard it twice and 3 times, so by the end of the day, you'll know, at least in some level, what I'm talking about. Are you awake? Excellent. Okay, well, thank you, and Misha, you're up.